Good day, everyone. I am Jillian Triana Bautista.
And I am Lozada from Philippine Science High School main campus, presenting to you our research entitled Comparing the Binding Properties of Dronabinol, Verubecestat, and Scyllo-inositol in Targeting the Amyloid-Beta Peptide through Molecular Docking.
Alzheimer's disease is a neurological disorder wherein brain cells progressively degenerate. This disease is the seventh leading cause of death, commonly affecting those aged 65 and older. The disease is due to the buildup of amyloid-beta peptide and tau protein in the brain. This causes the decrease in neurotransmitters, which are responsible for sending signals between brain cells. Studies have suggested that the drugs dronabinol, verubecestat, and scyllo-inositol can be effective in reducing the amount of amyloid beta in the brain. Dronabinol targets the protein acetylcholinesterase, which forms a stable complex with the amyloid beta protein. Verubecestat inhibits the beta site amyloid precursor cleaving enzyme 1, preventing amyloid beta peptide formation. And scyllo-inositol coats the surface of the amyloid beta peptide by binding with its protofibrils, blocking the development of amyloid beta. Through molecular docking, the binding properties of protein-ligand interactions can be determined to predict and compare the probabilities of binding occurrences.
The objectives of this study are to model methods of amyloid beta inhibition using molecular docking software as a supplement to future Alzheimer's treatments, to analyze binding properties of simulated interactions between potential inhibitors for acetylcholinesterase, base 1, and the amyloid beta peptide, and to identify amino acid residues and their effects on binding affinities and subsequently protein inhibition. Determining the binding properties of these drugs to the amyloid beta protein could aid in the advancement of Alzheimer's research. This will also give more knowledge about the selected ligands and proteins. Examining and differentiating the properties of the given receptors can potentially lead to drug discovery. The drug and protein models were obtained from PubChem and PDB respectively. For the preparation of the proteins, water molecules were removed from the protein, then polar hydrogen molecules and Coleman charges were added. A 3x3 docking matrix was performed wherein each ligand was docked to its target protein as well as the other two proteins. This is to study their interactions and determine whether or not the drugs would be able to inhibit the other proteins better than their target protein. A grid was prepared on the protein areas containing the active sites of the protein which were identified from literature. The docking portion was done on the command prompt using Autodock Vina and displayed different poses ranked from best to worst, as well as the calculated binding affinity. Interaction maps were then made using the protein ligand interaction profiler in order to study the interactions.
Dronabinol docked with its target protein acetylcholinesterase exhibited the best docking score. Verubecestat exhibited the highest binding affinity for its target protein base 1. And scyllo-inositol docked with its target protein amyloid-beta peptide produced the lowest binding affinity and the lowest overall binding affinity among the three drugs under study. These images show the drugs docked with their target proteins. They show the residues, which are specific binding sites on the protein, and the bond types, which entail the strength of the bonds. Hydrophobic interactions and hydrogen bonds were exhibited in the docked complexes. These intermolecular forces vary in strength. Hydrogen bonds are stronger than hydrophobic interactions.
Although hydrogen bonds are stronger than hydrophobic interactions, it is likely that scyllo-inositol was not able to inhibit the amyloid beta peptide well due to the lack of aromatic rings in its structure, which are important for hindering fibril formation and aggregation in amyloid beta. In conclusion, it is found that dronabinol and verubecestat may work well as potential inhibitors of their target proteins. However, scyllo-inositol may not be a good inhibitor for its target protein, amyloid beta peptide. Further research is required to test the effectivity of these drugs as Alzheimer's treatments.
And that is the end of our presentation. Thank you for listening.